Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 50, where I will discuss about the monolignols, lignins and lignans. So, the concepts covered will be biosynthesis of hydroxycinnamic acids, biosynthesis of monolignols, new view of monolignol pathway and lignins, and if time permits I will talk briefly on the lignans, otherwise lignan aspect will be covered in one of the subsequent classes. So, let us go to the next slide. So, in the previous class I have given an overview of phenylpropanol metabolism. This is just to make a quick revision that this started with phenylalanine, and by the action of the enzyme PAL, phenylalanine converted into transenamic acid, and transenamic acid is basically a C6C3 compound, and transenamic acid again converted into paracoumaric acid by an enzyme called cinnamate 4 hydroxylase. So, it acts 4th position OH and it forms 4 hydroxycinnamic acid or paracoumaric acid. After that, paracoumaric acid will get activated by addition of CoA. So, it is a CoA ligase. So, and then finally, it forms the different monolignols. So, our point of discussions today is about the lignin, and the precursor of lignin is the monolignol. So, monolignols are nothing but the precursor of lignin. So, that is our mostly the point of discussion, and this is basically the structure of hydroxycinnamic acids, which I have also shown in the previous class. So, for example, how the structure changes I would like to show you. This is called transenamic acid, and once OH is added, this is cinnamic acid or transenamic acid, and if one OH is added here, then it will become paracoumaric acid or 4-coumaric acid or 4-hydroxycinnamic acid, same word. So, now this structure can be further modified into by addition of one hydroxy group here. So, now this structure is called caffeic acid or 3,4 dihydroxycinnamic acid. From this, if you compare the structure between this coumaric acid and caffeic acid, then it is quite clear that a simple hydroxylation at 3 position of the benzene ring will convert 4-coumaric acid to caffeic acid. So, ideally one should say that there might be an enzyme called C3H, which may be a cinnamate 3 hydroxylase, which converts coumarate to caffeate, and lot of attention was paid on this, and initially it was thought to be a soluble enzyme, but much later when molecular tools were explored, then it was found that this conversion is not that simple. So, in other words, coumarate to caffeate conversion is not possible through this way. Okay. So, we will see then how this conversion will happen, but let us now go to the other structures. For example, this ferulic acid. Ferulic acid, this one was OH is basically the caffeate, Now, if you remove the OH and add CH3, so it will become ferulic acid. So, one thumb rule is this: in order to have the methylation, first there will be hydroxylation followed by the methylation. So, and ideally such conversion is possible. So, a methyl donor should be available and then it makes this one. Then subsequently there is another structure which was also elucidated. This is ferulic acid. Which is OH addition here. So, when OH added at 5 position, so it is called 5 hydroxy ferulic acid, and this 5 hydroxy ferulic acid can be converted into synapic acid, as you see the structure here. So, this at 5 position, now this is replaced with a methoxy group. So, this is 5 position, this is 6 position, this is 1, 2, 3, 4. So, this will become synapic acid. So, again there will be a donor for this methyl group. So, this is basically all the hydroxycinamic acids, and it was thought that hydroxycinamic acids act as a precursor for several phenolic compounds, including the monolignols. Now, hydroxycinamyl alcohols are the monolignols, because these are the precursors. One is the coniferyl alcohol, which basically coming from ferulic acid, because structure, if you remember the structure, it is this one, and the other one is basically the coumaryl alcohol, where only a OH group. So, which is basically originated from coumaric acid. So, this is for coumaryl alcohol or paracoumaryl alcohol or the same, and the synapyl is basically originated from synapic acid. So, the structure as you see here. So, this is the synapyl alcohol. So, these three are the three major monolignols that contribute to the formation of lignin. Okay. So, the overview of aromatic metabolism in plants, what I have started talking from the last class. So, phosphenol pyruvate erythrose 4-phosphate that joins by the DHP synthase, which produce DHP, and I have mentioned that this is a very active step, because this is a regulatory step, and eventually via secchymate it forms chorismate. From chorismate, the one route goes to tyrosine, other route goes to phenylalanine, and there is another route which goes to tryptophan. Okay. So, and phenylalanine is basically the starting point for phenylpropanoid metabolism, although tyrosine can also be the starting point, particularly for the monocots. And we have also mentioned about the PAL enzyme, which is phenylalanine ammonia lyase, which releases the ammonia from here and converting phenylalanine to cinnamic acid. So, this cinnamic acid usually trans-cinnamic acid, and this subsequently by the action of cinnamate 4 hydroxylase it forms paracoumaric acid, and subsequently other hydroxycinnamic acids. This is a common structure, and this step is very important, which is the activation of hydroxycinnamic acid by the 4-Cl. I have also mentioned this. So, this is basically addition of CoA thioester, and this is now the activated compound. From there different route emerges. For example, the route towards flavonoid and anthocyanin moves in this way, the route towards lignin which moves in this way, the route towards other phenylpropanoid that moves in this way. So, specific branch points. So, this is general overview of the aromatic acid metabolism, and then now we will I have already mentioned about these structures. If you that paracoumaryl alcohol I have mentioned just in the previous slide. So, which is one of the monolignols, and the lignin which derived from paracoumaryl alcohol dominated or the lignins which is basically dominated by the paracoumaryl alcohol are called para-hydroxyphenyl lignin, or in brief it is called H lignin hydroxyphenyl. Okay. Whereas, the coniferyl alcohol which is basically coming from the ferulic acid, as you see the structure here. So, coniferyl alcohol that actually contributes to the formation of guaiacyl lignin, GUAIACYL, guaiacyl lignin, or it is called G lignin. Whereas, the third one is the one which derives from synapate, or the synapyl alcohol is the monolignol that makes the syringyl lignin residue, or it is called S lignin. Now, one point I must mention here that H lignin is basically dominated in the monocots. Example is grasses. Whereas, the G lignin is found to be the only lignin in the conifer, that is gymnosperm. So, conifer lignin is only the G lignin, whereas S lignin you will find in angiosperms. So, angiosperms you will get a fine balance between S and G. So, a balance between S also you will find some H lignin is also present. So, in monocot although H is dominating, but you can also get little bit of S and G, but normally in the angiosperm it is S and G, and that actually makes a lot of issues, particularly in terms of delignification, in terms of lignin quality, all these things. So, maybe I will discuss a little bit later. So, now this is what I am showing, this is basically a old scheme or the classical scheme of monolignol pathway. So, now this scheme is not fully valid now, but it is important that we should know this, and then we will appreciate the what are the new discoveries made on this. So, this was basically published in the first year of the journal which is called Trends in Plant Science. So, Trends in Plant Science is basically the journal which started publishing in 1996. Okay. So, what we see here is again the cinnamic acid. By the action of cinnamate 4 hydroxylase, it makes paracoumaric acid. Then C3H, you see that C3H is mentioned here, because see that it is as early as 1996. So, still at that time there was an argument that in fact the C3H exists, it may be a soluble enzyme. So, that is why, but now what I have mentioned that as such C3H is not functioning nowadays. So, caffeic acid can convert it into ferulic acid by a methyl transferase. This is possible, okay, because you know that this added, and then ferulate 5 hydroxylase basically converts ferulic acid to 5 hydroxyferulic acid at this position, that is all right, and then subsequently the COMT which makes this position methyl group added. So, these are all 5 hydroxycinnamic acids I have already mentioned. Now, this coumaric acid by activated by making paracoumaryl CoA and contributing to paracoumaryl alcohol, whereas the ferulyl CoA which originates from ferulic acid contributed to coniferyl alcohol, and the synapyl CoA which originated from the synapic acid now contributed to the formation of synapyl alcohol, and I have mentioned about the respective lignins formed by these monolignols like H, G and S. So, here this scheme is pretty straightforward. So, what you see here, that basically there are 3 parallel pathway operating, this one, other one is this one and this one, and the intermediates like caffeate and hydroxyferroyl alcohols, that is not directly involved in the monolignol formation, perhaps it supplies the intermediates from one branching point to other branching point. So, methylation pathway. So, methylation is very important and there is no wrong in the methylation discoveries. So, now what is known? What is now known that the Kumaric acid, so I better use this whiteboard, that structure I will show in the next slide, because drawing structure will take time, I will not finish in time. So, 4-Kumaric acid or para-Kumaric acid cannot directly convert into caffeic acid, but it is first converted into para-Kumaroyl CoA. This para-Kumaroyl CoA subsequently converted into either para-Kumaroyl sikimate or para-Kumaroyl caffate. So, it is joining with a sikimic acid. So, sikimic acid biosynthesis I have shown in the previous class, and also apart from sikimic acid, quinic acid can also join, because that also I have mentioned in this quinate sikimate pathway. So, here for the sake of our discussion we will concentrate only on the sikimate. Quinate issue I will discuss maybe in the next class when we will talk about other caffeic acid esters. So, para-Kumaroyl CoA joins with the sikimic acid and it forms the Kumaroyl sikimic acid or para-Kumaroyl sikimic acid. So, this para-Kumaroyl sikimic acid will be, actually I should make it in that color, otherwise there is no point of. So, this converted into caffeoyl sikimic acid. So, the caffeoyl sikimic acid and then this caffeoyl sikimic acid, what will happen from here, this leads to the formation of caffeoyl CoA, leaving the sikimic acid out. So, basically there are 3 reactions. The first and third reaction are catalyzed by an enzyme which is called HCT, hydroxycinnamoyl CoA, this is cinnamoyl caffeoyl transferase. So, it is basically a transferase which basically joins sikimic acid and Kumaroyl CoA make para-Kumaroyl sikimic acid, and the next enzyme is basically the C3H, which converts para-coumaryl secumate to caffeoyl. So, remember that coumaryl and caffeoyl is what is the difference between the structure. If I briefly tell, that is coumaryl only 4 position and caffeoyl addition of a hydroxylase at 3 position. So, this is caffeoyl and this is para-coumaryl. So, this addition is basically this is addition. So, which means a hydroxylase. So, C3H is nothing but a 3 hydroxylase, it added 3 position which converts caffeoyl coumaryl to caffeoyl. So, and then finally again the HCT which converts caffeoyl secumate to caffeoyl CoA, and this caffeoyl CoA subsequently will be utilized by another enzyme leading to the formation of another important structure which is called ferulyl CoA. So, when it is ferulyl CoA, then basically there will be a CCOMT enzyme will be there. Caffeoyl CoA O methyl transferase, what will happen that this position OH will be replaced with a methoxy. So, that is why OMT working here. I think it is clear to you. It will be more clear in the next slide when I will see the structure. So, the take home message is that coumarate to caffeate conversion is not simple. It involves a complex route using either secumate or quinic acid and making para-coumaryl secumate that converted into caffeoyl secumate. Here C3H plays important role and then it converted again back to caffeoyl CoA. So, this is now clear here in this, as you see here that this is para-coumaric acid. So, para-coumaric acid first with the 4-Cl enzyme it makes para-coumaryl CoA, that para-coumaryl CoA now joining with the secamic acid and here is the HCT and which makes the para-coumaryl secamic acid, and then it is the C3H what I have shown. So, you see the structural difference as I have mentioned in the previous slide that. So, this is only OH, when is a caffeoyl, then OH added, additional OH added here, this is additional, that is why C3 prime H is C3H is there. Okay, and then this converted into again by HCT which makes caffeoyl CoA, and I have said that caffeoyl CoA subsequently utilized in making the ferulyl CoA and the pathway moves. However, caffeoyl CoA can also form from caffeic acid by another 4-Cl reaction. Okay. So, that means this is what that coumaryl CoA to caffeoyl CoA is a multi-step reaction, this part that is called meta hydroxylation, which is the complexity of the lignification pathway. So, this was all discovered in 2004, 5, 6 that time onwards. So, you will first see such pathways coming into the publications much later in the book. So, from 2006 onwards in the publications. So, one such publication I have referred somewhere. Okay. And then you see that the H unit, no problem with H unit. For the G unit, that means G unit is basically moving in this way. Okay. And the S unit is basically moving in this way. That means the synapaldehyde, coniferaldehyde, coumaryl, para-coumaryldehyde, how it is formed? It is formed from para-coumaryl CoA. Coniferaldehyde, how it is formed? It is formed from ferulyl CoA, and the enzymes that catalyzes those reactions are this CCR, cinnamyl CoA reductase, a generic name. Okay. So, and Okay, so that means the ferulic acid is not directly involved in making ferulic acid is not directly involved in making ferulyl CoA, and the synapic acid is not directly involved in making this synapoyl CoA. That means this is not usually not functioning. So, function is performing in this way. So, this is again a new discovery, and this is basically how this once the monolignols are formed, they basically moves out from the cytosol to the cell wall space, and then there lies the enzyme lactase and peroxidase, and then it forms different linkages, beta O4 linkages, 4O5 linkages, 5,5 linkages, and then they form the detailed polymer. So, the so this is basically the papers which published in 2000 and 2001 in two very high impact journals. One is journal Biological Chemistry and Plant Cell. So, there they have mentioned these things. So, the first one is basically talked about these two steps. So, that means coniferaldehyde converted into synapaldehyde, and the second one is basically talks about there is an existence of SAD. So, and which converts this to this. So, now this will be clear here. This is a simplified diagram. So, what I have shown again just to make the concept clear, parakumaroyl CoA making caffeoyl CoA, but in order to make this you require HCT 2 times and C3H. Okay. And so, basically what is here HCT C3H again HCT, then it makes caffeoyl CoA, then caffeoyl CoA methyl transferase, basically OM methyl transferase, which makes ferulyl CoA. Ferulyl CoA by the action of CCR, cinnamoyl CoA reductase, which makes coniferaldehyde, and coniferaldehyde by the action of CAD, which makes coniferyl alcohol, that is one of the monolignol which converted into, which is contributing towards the G lignin. Whereas, the new pathway says that from coniferaldehyde synapaldehyde is formed. Synapaldehyde is not from the synapate. So, through the enzymes called this CALD-OMT, which is F5H is basically named as this CALD5H, coniferaldehyde 5 hydroxylase, CALD5H, and the second one is called ALD-OMT, aldehyde O methyl transferase, which convert one aldehyde to another aldehyde. 5 hydroxy coniferaldehyde converted into synapaldehyde by a methyl transferase. So, the reaction is like this. This is one, five hydroxy coniferaldehyde converted into synapaldehyde. So, OMT means O methyl transferase, as you see here, that here is the addition and here this OH, but here it becomes OCH3. Okay, and then by the action of CAD and SAD, synapyl alcohol dehydrogenase. So, both CAD and SAD can work, and this makes synapyl alcohol, and then they are transported to the cell wall, and oxidative polymerization takes place by the action of peroxidase and lactase, and ultimately it forms the lignin. So, this is basically the revised model. So, what I have said, just briefly go through it, so that it will settle in your mind. So, kumaric acid, kumaroyl CoA, from there it moves to parakumaroyl secumate to caffeoyl secumate, and then it comes back to caffeic acid. Then from here it goes to ferulic acid, then coniferaldehyde, one route goes to coniferaldehyde to coniferyl alcohol, and the route for the synapoyl goes in this way, and then eventually it forms the synapaldehyde and synapyl alcohol. So, this is basically the joining of different scheme leading to the formation of different monolignol. So, this is another way of representation. So, this was actually first appeared in environmental chemistry letters by a group, and I picked up this one that time, and then with further addition we make it. So, here basically they have mentioned all the enzymes like C3H or C3 prime H. So, that is converting this coumaryl to caffeoyl, which is called coumaryl ester 3 prime hydroxylase. So, both C3H and C3 prime H is used. C3H is used when you are talking about the coumaryl secumate to caffeoyl secumate, but when you are talking about the coumaryl quinate to caffeoyl quinate, there C3 prime H is used. That is what a normal convention. Okay. And finally, that apart from this H lignin, G lignin and S lignin, recently there are arguments and evidences that C lignin, which is from the caffeoyl, it is a possibility that that can also be formed, and although these steps are unclear, but from caffeoyl CoA, as caffeoyl CoA contributes to ferulyl CoA. So, caffeoyl CoA can by the action of CCR can make caffeoyl aldehyde, and caffeoyl aldehyde by the action of CAD can make caffeoyl alcohol. So, this is nothing but the structure of caffeoyl alcohol. So, that may contribute to the C lignin, and other one is this, that the ferulate 5 hydroxylase or which is the intermediate which contributes to 5 hydroxyferulate, or this is or 5 H we can also call this CALD this CALD 5 H. So, that I have mentioned previously. So, this ultimately makes what? This is the structure of 5 hydroxyferulyldehyde. 5 hydroxyferulyldehyde is already produced in the pathway, and this 5 hydroxyferulyldehyde simply one requires only a specific CAD, which converts 5 hydroxyferulyldehyde or coniferaldehyde into this 5 hydroxy coniferyl alcohol. This is 5 hydroxy coniferyl alcohol. This is the coniferyl alcohol. This is coniferyl alcohol, and this is 5 hydroxy coniferyl alcohol. So, simply one CAD can convert the already this coniferyl aldehyde, 5 hydroxy coniferyl aldehyde is already produced by CALD 5 H, coniferyl aldehyde 5 hydroxylase. So, this is produced. So, this can move in another direction, and it eventually may contribute to 5 hydroxy lignin, which is called 5 H lignin. So, these are the new view, and this was published in Trends in Plant Science in the year of 2018. So, this is a review appeared in 2018. So, I have given here, but anyway I did not give you the detailed references. So, this is all about the classical and contemporary views of monolignol pathway. Okay, lignans I will not discuss, I will discuss in a separate class. With this I end this class. Thank you very much.